Hello everyone, welcome back to Microbial Chronicles. In today's video, we are going to learn more about the role of each component of gram staining, the principles of gram staining, and the examples of gram positive as well as gram negative bacteria. In the previous video, we learned about the procedure of gram staining in detail and what are the components required for it. So today we are going to learn about more in detail about each of these things. So now the first component, as we learned last time, was heat fixation or any fixation. So the use of this fixation is that it prevents displacement of the smear and also the internal structures of the cell are protected. So that is the main function of fixation of the smear. Next will be the primary stain, which in most of the cases is crystal violet, but also methyl violet or gentian violet can also be used. So the main function of this primary stain is that it stains all the bacteria in the field as violet. Irrespective whether it's gram negative or gram negative bacteria, all the bacteria will be seen as violet. Then comes the mordant. So the mordant was grams iodine. Now the function of this mordant is that it binds to the dye to form bigger diiodine complexes. So this mordant, which is gram iodine, binds to the primary stain, which is crystal violet, to form bigger diiodine complexes. Then comes the decolorizer. So the use of the decolorizer is very important, as I already discussed in the last video, which is the most critical step. Now I'll explain why it's critical. Because what it does is that it removes the primary stain from the gram negative bacteria, whereas the gram positive bacteria retains the primary stain. But it's important to note that if the decolorizer is kept for a longer time, that's why I stress that the timing is very important. If the decolorizer is kept for a longer time, it can decolorize even the gram positive bacteria. Therefore, your gram positive bacteria will appear as gram negative, and that can lead to an error in diagnosis, which is wrong. Lastly was the counter stain, which imparts a pink color or a red color, so which is saccharin. So now we move on to the principles of gram staining. So we have three theories: pH theory, cell wall theory, and a magnesium ribonucleotide theory. But I will only discuss pH theory. So just to understand what happens over here, cytoplasm of gram positive bacteria is acidic, and the dye, which is crystal violet, is basic. And this acidic cytoplasm therefore retains the basic dye more strongly. And the iodine is the mordant which combines with the primary stain, and therefore forms a diiodine complex which gets retained inside the cell. So this is the simple pH theory. So now we'll move on to the cell wall theory. So as we already discussed in the previous video about the gram positive, gram negative cell walls, the gram positive bacteria have a thicker cell wall. So basically they have a thick peptidoglycan layer, which means that they have tight cross linkages. So therefore, because of tight cross linkages, the peptidoglycan prevents loss of the primary stain. And the alcohol which they added shrinks these pores further, so makes them even smaller. And as we already discussed, the diiodine complex is larger, so obviously that will not be able to penetrate and not go out. This is why the gram positive bacteria will retain the primary stain. So just to reiterate, tight cross linkages therefore prevents loss of primary stain, and alcohol shrinks the pores even further, and large diiodine complexes cannot penetrate this. So this is the main theory for gram positive bacteria. Now I'll discuss about gram negative bacteria. So gram negative bacteria has a thinner peptidoglycan layer. Okay, so it's a more permeable cell wall which allows the outflow of the primary stain because of the thinner peptidoglycan layer, and this is not tightly cross-linked. And also the lipopolysaccharide layer is disrupted more easily by decolorizer and forms larger pores. Just to quickly recap this, gram negative bacteria has a more permeable cell wall which allows outflow of the primary stain, and thin peptidoglycan layer does not have tight cross linkages, and the lipopolysaccharide layer is easily disrupted. So therefore this is the cell wall theory. So this is the most important theory, arguably, between the two theories. And magnesium ribonucleotide theory is I will not be discussing over here. It's a little more advanced theory. So I will not discuss it right now in this video. Now we will move on to the types of gram-positive bacteria and the types of gram-negative bacteria. Whether the bacteria is gram-positive or gram-negative, they can be divided into cocci or bacilli. So bacilli means rods. The cocci, and now let's first discuss about the gram-positive cocci and gram-negative cocci. So the gram-positive cocci, they are like means spheres. Cocci is like sphere or circle. Okay, so now gram-positive cocci arranged in clusters. Example is Staphylococcus. Okay, in the same cocci, if they arrange in a long chain, example will be Streptococcus. And the gram-positive cocci arranged in pairs of Diplococci, example will be Pneumococcus. So this is about gram-positive cocci. And there are again various arrangements tetrads and octads, but I won't be discussing that over here. Now gram-negative cocci, mainly we have meningococci which is lens-shaped and gonococci which is a kidney-shaped, which is visible over here. Now lastly, we have the gram-positive bacilli and the gram-negative bacilli. So gram-positive bacilli examples are Bacillus anthracis, which shows a bamboo stick appearance, and the Corynebacterium diphtheria, which was a Chinese letter of cuneiform arrangement. Lastly, the gram-negative bacilli. So there is examples would be a simple rod would be members of family Enterobacteriaceae, Klebsiella, Escherichia coli, Salmonella, Shigella, etc. And the other gram-negative bacilli we have is Vibrio cholerae, which is comma-shaped gram-negative bacillus. So it has a school of fish appearance. So these are at least some of the basic bacteria which we can see with gram staining, and many other examples, but I just covered the main ones over here. So thank you everyone, and we've come to the end of today's video. I hope you all have understood this concept. If there are any queries, you can please comment it. So don't forget to like this video, share and subscribe if you found this helpful, and I will see you all in the next video. Thank you.